Hi folks, I'm back for part 2 of the third lecture on coronavirus. In part 1 I described to you how the coronavirus RNA is copied inside of cells to produce many new copies of the coronavirus genome that can be used to assemble new viral particles. In this second part, I'll show you how the coronavirus proteins are produced so that you can make many copies of those coronavirus proteins, which can then go to assemble with the RNAs and to yield intact coronavirus particles that are then released from cells. All right, so where we left off last time is we had a cell, and inside the cell we had the coronavirus positive strand RNA, and I'm going to make one change now. I'm going to show the negative strand in brown so it's easier to distinguish it. And the positive strand RNA was replicated to make a negative strand RNA. And then that negative strand RNA is separated from the positive strand, and it gets copied multiple times. Each time it's copied, it generates a new positive strand. So you have, a whole bunch of positive strands that can be produced from that negative strand. And these will then be used to assemble new coronaviruses in the future. Now, how do the proteins get produced? Well, this positive strand, as I showed you, it gets translated to make a very long protein that's chopped up into 16 different proteins. And a group of those proteins are used to make this negative strand. I already described that. But you still have to make the proteins that are in the envelope, the S protein, the E protein, and the M protein, as well as the N protein, the nucleocapsid protein. So those haven't been produced yet. So let's take our positive strand here, and after making the long negative strand, the virus, and making the proteins that'll enable the copying, the virus then makes short negative strands, and it makes a bunch of them, and I'll just show you 4 of them. So let's make one here, and it makes another one here, and another one here, and another one here. And these negative strands can then be used to produce positive strands, the same way this process works here. So you end up with, 1, 2, 3, 4. Now these positive strands can be translated to make proteins, just like the original positive strand was translated to make that very long protein that got chopped up into 16 other proteins. Now this one would code for the S protein, the E protein, the M protein, and the N protein, okay? So this would be S, E, M, N. The problem is the virus has to make a lot of those proteins. The first long protein, it didn't have to make a lot. Here it's got to make a lot because it's literally got to manufacture. These are the actual physical structural components of the virus. And so you need tons of those. Now, the problem is, so the cell has a lot of ribosomes. So I'll draw some ribosomes here. And these are, all over the cell. But those ribosomes, they're not sitting there waiting to be infected with a virus and to make the viral proteins. They're happily translating RNAs inside the cell. So these would be all the RNAs that the cell needs to make its own proteins. And so this is going to make it hard for the virus to actually command enough of the factory output, shall we say, of the ribosomes to make all the proteins that it needs to make. So the way the virus deals with this is one of the proteins that was made early on from the positive strand genome. Remember, it made that very long protein, it chopped into 16. One of those 16 proteins goes around and it shuts down the ability of the cell to start making new proteins of the cell's own proteins. But even more nefarious than that, it recruits other factors in the cell, and we still don't fully understand all of this, and those other factors chew up these RNAs for the cellular proteins. So they're essentially eliminated. So now the cell can't make its own proteins because its RNAs have been chopped up. So these ribosomes can now be redeployed onto the viral RNAs. So the viral RNAs can now come in and essentially commandeer these ribosomes. So this could be the S1, for example, the E1, the M1, and the N RNA. And these get translated many, many times to make large amounts of the S, E, M, and N proteins. And that will then enable this, the assembly of many, many new copies of the virus. So for the last part of this, I drew it ahead of time because the drawing gets a little bit complicated. Once you have all those copies of the S, E, M, and N proteins, they begin to assemble. And that's diagrammed here. So as I told you in a prior lecture, the N protein, the nucleocapsid protein, assembles with the RNA and it packs the RNA, so that would be these RNAs here, into a very, very tight structure so that it can be packaged into this tiny little capsule of the virus. Meanwhile, the E, M, and S proteins, those are all membrane proteins. And so when they're synthesized, they're inserted into membranes inside the cell in the same way that when the cell makes proteins for its own membranes, it inserts them into the membranes as it's making them. So the virus uses, it piggybacks on the same mechanism that the cell is already using to make its own proteins. So here we have the virus has commandeered the ribosomes, the E protein, M, and S proteins that have been produced are inserted into this membrane. This is the cell here, so this is all happening inside the cell. And these envelope proteins now recruit the RNA and nucleocapsid, and this whole assembly starts budding inwards in much the same way that I showed you yesterday for how the virus enters into the cell from outside the cell directly through the membrane of the cell. That was the first entry pathway I showed you. So it buds inwards into this membrane structure in the cell, and you end up with the virus like this. You end up with a free virus inside this membrane structure. And I forgot to draw the RNA in here, but imagine it's in there. So now this structure can fuse with the membrane of the cell, and when it fuses, if you can imagine this happening here, and I'll just draw it out. What happens is now these components can get released to the outside of the cell. And so that's how the virus, that's how you make new copies of the virus and release them from the cell. One thing I forgot to mention is as all this is going on, so for this process, you need 3 of the 16 proteins that are cut from that long protein. 3 of them are involved in this process of actually forming the virus, but they're not incorporated into the virus. They help assemble, they're like scaffold proteins that are not part of the final structure, just like scaffolding on a building is not part of the final structure. And then there's another couple of proteins of those 16 that are thought to aid in the release of the newly formed virus from the cell so that it can go past to other cells. And so that completes the third lecture on how coronavirus, once it gets inside a cell, copies its genome and makes new proteins by commandeering the cell's synthetic machinery so that it can assemble many new viral particles to be released from the cell. And in the next lecture, I'll begin discussing how the host responds to the presence of this viral infection. In particular, how the immune system detects and responds to this invasion. Thanks for your attention. Have a good day.